Welcome to the video of biochemistry methods. In this video, we will learn about centrifugation, electrophoresis, and chromatography. Let's start with centrifugation. Centrifugation is a group of separation techniques in which a centrifugal field is used. It's used to separate mixtures of biological components in a liquid medium. For example, if we want to remove or separate the solids in a liquid solution, we can centrifuge them, and in the end we will obtain the precipitate with the solids and on top the liquid supernatant. Therefore, centrifugation speeds up the natural process of sedimentation by gravity. Now, let's understand the factors that influence centrifugation. For that, we have to know that there are three forces that act on particles in solution during centrifugation. We have the centrifugal force that pushes away the particle away from the rotation axis. And then we have the buoyancy force and the frictional force that opposes to that movement. So the centrifugal force depends on the mass of the particle and on the centrifugal field. That is the angular velocity of the rotor squared times the distance from the center of the rotor. Then we have the buoyancy force. The buoyancy force is the force that the particles that sediment do for displacing the liquid that is around them. And this force depends on the mass of the solution displaced by the particle and again by the centrifugal field. Finally, the frictional force depends on the sedimentation velocity of the particle and on the friction coefficient that in turn depends on the medium viscosity and on the radius of the particle. So at constant velocity of the rotor, the sum of all forces will be zero. Then, solving for the velocity of the particle, we obtain this equation. And then if we isolate the sedimentation coefficient, we can see that the sedimentation coefficient is just the moving velocity of the particle per unit of centrifugal field. Looking at the formula, we can see that the sedimentation coefficient depends on the medium, exactly about the density and the viscosity of the medium, and on the particle, on the density and on its shape. So heavier particles will have higher sedimentation coefficients and will sediment faster. So usually the centrifugal field is expressed as a multiple of the air gravitational field, the relative centrifugal field. The RCF is adimensional, but it's commonly expressed in G's, that is the number of times higher than the gravity. So there are different centrifugation modes. We have differential centrifugation, in which initially the mixture components occupy the whole tube. And after centrifugation, we obtain a pellet with the components with higher sedimentation coefficient and a supernatant with the rest of the components. This mode can be used for cell fractionation, for obtaining different organelles of the cell by doing different rounds of centrifugation with increasing speeds of centrifugation. Another mode is the density gradient centrifugation. In this mode, usually not always, the mixture of components is applied on top of the tube, and then the mixture components during centrifugation will be separated in bands or zones, as you can see in this picture. For obtaining this, we need a density gradient along the tube, otherwise we will have distortions that will affect the separation. Commonly, substances for obtaining these gradients are, for example, glycerol or saccharose. Finally, analytical ultracentrifugation consists in that we apply a very high centrifugal field, and this can be used to determine the properties of the molecules. Now let's talk about electrophoresis. Electrophoresis is a separation technique in which charged molecules are separated when they are exposed to an electric field. The main steps are preparing the support. The support will be in this electrophoretic tank, and it will be embedded with the buffer. Then we will apply the sample and a marker with no molecular weight, and we will start the current. Then the separation can begin. Finally, after the separation, we have to stain the molecules to make them visible. So important factors in electrophoresis are the support material. The support doesn't have to have any charge and doesn't have to absorb the material on it. There are also two main types of support, ones that are non-restrictive in size and others that are restrictive in size. The ones that are restrictive in size provide a better resolution. Then about the electric field. Increasing the voltage will increase the velocity of the molecules, but at the same time will increase the temperature and then can create distortion. So we have to make a trade-off. And finally, the buffer is very important because we want to keep the pH constant. So for an ideal spherical particle with charge Q in a liquid medium under the influence of an electric field E, this particle will be subjected to two forces, the electric force and the friction force. So when we start the electric force, the velocity of the particles will increase, but this increasement in the velocity will also increase the friction force. Then soon the friction force will equal the electric force, and solving for B we have this equation in which we can see that the velocity of every particle depends on its own properties, its radius, and its charge on the medium and of the electric field. So for protein electrophoresis, we use as a support polyacrylamide. That's why it's called polyacrylamide gel electrophoresis, or PAGE. Polyacrylamide acts as a molecular sieve, so proteins will be separated as well by its size. And then we have two modalities, electrophoresis in denaturing conditions, also called SDS PAGE, because we use SDS, that is a detergent that denatures proteins and confers them negative charge. So, since all proteins will have the same density of negative charge, the separation will only be because of its size. And then we also have electrophoresis in native conditions. In this kind of electrophoresis, the protein will remain in its native 3D structure. This allows the study of conformation, aggregation, and the binding to other molecules. And then for the electrophoresis of nucleic acids, we use as the support agarose. Agarose is a polymer that forms a microporous structure. Therefore, these nucleic acids will also be separated by size. And only by size because the nucleic acids, all of them have the same density of negative charge. Therefore, the smaller fragments will run faster through this microporous structure. In addition, plasmids, since they have three conformations according to the level of supercoiling, they will run at different velocities every conformation. So for example, we have here a sample of a plasmid, the same plasmid. And depending on the conformation, we have here the supercoiled form that will run faster, then the linear form, and then the circular form. Now let's talk about chromatography, and in special about liquid chromatography. So chromatography is a technique for the separation of components in a mixture. Every chromatography has two phases, a mobile phase that dissolves the component of the sample and carries them through the stationary phase, and a stationary phase that is a static phase that interacts with the components of the mixture. The separation is due to the different partitioning of the components of the mixture between the mobile and the stationary phase. Therefore, the stronger the interaction of the solutes with the stationary phase, the longer will be the retention time. So there are different separation principles in chromatography, and according to these separation principles, a classification has been done. So one separation principle can be solubility. For example, if we have as a stationary phase a polar component, then polar solutes will be retarded in comparison to no polar solutes. Why? Because of the like dissolve like principle. Another separation principle is adsorption to a solid stationary phase. For adsorption, we can be due to electric charges, hydrophobic interactions, specific biological interactions, or to other inspecific interactions. And finally, another separation principle is the size. Then we can have, depending on the geometry of the stationary phase, planar chromatography or column chromatography, depending if it takes place in a planar surface or in a column. For planar chromatography, the main separation principle is solubility, depending in the solubility of the components, for example in paper, to the cellulose groups. And in column chromatography, the main steps are setting up and equilibrating the column, then applying the sample. We have to remember that the mobile phase will be running all the time. And then the different components will be eluting from the column at different retention times. But for the components that might be adsorbed in the column, we will have to elute them by changing the conditions. And then, of course, the different fractions can be separated by fractioning. So in column chromatography, it's important to understand the chromatogram. So the retention time is the time that it takes to a solute to go out of the column. The dead time is the retention time of a solute that does not experience any retardation at all. So for two compounds with different retention times, then we will be able to separate them in chromatography. Finally, resolution is the ability to separate two solutes. It depends on the selectivity and on the efficiency. Selectivity is the degree of separation between two peaks, while efficiency is the ability of the column to produce narrow peaks. So for example, here we have the perfect case in which we have very good selectivity and good efficiency. But here, for example, is one example of bad selectivity and here of bad efficiency. And here bad everything, bad selectivity and bad efficiency. And finally here we have the formula for the resolution, that is nothing more than the difference between the retention times of the two compounds divided by 1/2 of the sum of the width of the peaks of the two compounds. And now, thank you for watching.